The endothelium is a monolayer of epithelial cells that form a continuous lining along the inner surface of blood vessels and lymphatic vessels. It serves several major functions. Firstly, it provides a barrier between blood and the interstitial space of tissues, controlling the movement of molecules between them. It maintains the integrity of the blood vessel wall and helps to prevent blood from clotting. But here we will explore another role, to regulate the contraction of smooth muscle cells in the blood vessel wall, which control vascular tone. The first indication that the endothelium was more than just a barrier, keeping the components of blood in the lumen of blood vessels, came in 1980, with a serendipitous discovery in Bob Furchgott's lab in the USA. They found that arteries responded very differently to acetylcholine if the endothelium was removed. This trace, from their landmark paper, shows the constriction of intact aorta when norepinephrine was applied. When acetylcholine was applied to the constricted vessel, it reversed the constriction, so the vessel dilated. The trace below shows the same experiment performed on aorta after the endothelial layer of cells had been removed. This time, when the same concentration of acetylcholine was applied, there was no effect, but the constriction was reversed when the tissue was washed to remove norepinephrine. So the presence of the endothelium was necessary for acetylcholine to dilate the artery. The authors concluded that the endothelium releases a factor that relaxes vascular muscle, and they called the factor endothelium-derived relaxing factor, or EDRF. Subsequent research found that the factor was nitric oxide, which is a simple diffusible gas. These discoveries led to the 1998 Nobel Prize in Medicine, which was awarded to the main groups that identified the critical role that the endothelium plays in regulating blood vessels. Many agents acting on G-protein coupled receptors can activate the endothelium to release nitric oxide. Two examples are acetylcholine, acting on muscarinic receptors, and bradykinin, acting on bradykinin receptors. When either of these agents binds to and activates their receptor, they evoke a rise in the concentration of calcium ions in the cytoplasm of the endothelial cell. Another stimulus that raises calcium and activates endothelium-dependent dilation is shear stress, which is the frictional force on endothelial cells caused by flowing blood. Whatever the stimulus, calcium activates the cytoplasmic enzyme nitric oxide synthase, or NOS. There are three varieties of NOS, but this one is the constitutive NOS. NOS stimulates the synthesis of nitric oxide from its precursor arginine. Nitric oxide readily diffuses from the endothelial cell to the smooth muscle cell, and in the cytoplasm of the smooth muscle cell, it activates the enzyme soluble guanylate cyclase. The action of this enzyme is to stimulate the synthesis of cyclic GMP from GTP. Cyclic GMP activates a number of processes that together promote relaxation of the smooth muscle cell. Endothelial cells can also make an inducible form of NOS. Like the constitutive NOS, the inducible form also stimulates nitric oxide synthesis from arginine, and nitric oxide diffuses to nearby smooth muscle cells, where it stimulates guanylate cyclase to produce cyclic GMP. The same downstream events mediate muscle relaxation and vasodilation. This pathway is a major mediator of inflammatory responses, and in septic shock it produces profound vasodilation, which results in a large drop in blood pressure, which can be fatal. Inducible NOS is not normally expressed in endothelial cells, but the cells express receptors for cytokines, which are released during inflammation. When activated by cytokines, these receptors activate pathways that lead to the induction of nitric oxide synthesis. The inducible NOS is not activated by calcium. Instead, as new enzyme is expressed in the cell, it continuously produces nitric oxide in an uncontrolled way. The only way to inhibit the activity of inducible NOS is to prevent its synthesis with anti-inflammatory glucocorticoid drugs. Cytokine receptors are also expressed in smooth muscle cells, where cytokines cause additional synthesis of iNOS and further production of nitric oxide in the vessel wall. That is also blocked by glucocorticoids. Cyclic GMP, generated by nitric oxide stimulation of soluble guanylate cyclase, activates a cyclic GMP-dependent protein kinase G1. The protein kinase phosphorylates a number of key proteins involved in the regulation of muscle contraction. As a consequence, it stimulates SERCA, the calcium-pumping sarcoendoplasmic reticulum ATPase. This facilitates calcium accumulation by the sarcoplasmic reticulum, so that calcium is moved from the cytosol to the SR lumen. Protein kinase G also stimulates the activity of the plasma membrane calcium pump to extrude calcium from the cell. It also inhibits calcium channel activity, thereby reducing calcium entry into the cell. These effects all lower the cytoplasmic concentration of calcium. Protein kinase G also phosphorylates and activates myosin light chain phosphatase, causing dephosphorylation of myosin and muscle relaxation. Cyclic GMP can also stimulate the activation of large conductance calcium-activated potassium channels, which leads to potassium efflux and membrane hyperpolarisation. That, in turn, inhibits calcium entry through calcium channels, adding to the relaxation. Cyclic GMP levels depend not only on its synthesis, but also on its metabolism, which is mediated by phosphodiesterase enzymes. There are multiple phosphodiesterase enzymes, and drugs are available to inhibit particular types of these enzymes. One example is sildenafil, which inhibits the phosphodiesterase 5 isoform, resulting in the accumulation of cyclic GMP in the cell cytoplasm and muscle relaxation. As a consequence of this action, PDE5 inhibitors are used to treat erectile dysfunction and pulmonary hypertension. The effects of nitric oxide on smooth muscle cells can be mimicked by a group of drugs known as the Nitrovasodilators. All of these drugs are nitric oxide donors. In other words, they release nitric oxide inside the smooth muscle cell, where it activates the soluble guanylate cyclase in the same way as endogenous nitric oxide. These three drugs are all used clinically. Glycerol trinitrate is subject to extensive first-pass metabolism, so it is administered under the tongue as a spray or tablet, from where it is directly absorbed into the circulation. The release of nitric oxide from glycerol trinitrate requires the actions of intracellular enzymes. It acts quickly, but tolerance develops quickly too, so its effectiveness is limited. Its therapeutic action results primarily from a particularly potent action on veins. Isosorbide dinitrate and nicorandil are orally active nitric oxide donors with a prolonged action. All three drugs are used clinically to treat angina. Nitroprusside is a potent nitrovasodilator, but it decomposes when exposed to light, and unless freshly dissolved before use, it can produce toxic breakdown products. Its use is reserved for hypertensive emergencies. Amyl nitrite is a simple nitric oxide releasing molecule which is active when taken by inhalation. Due to the route of administration, it has a greater effect on arteries relative to glycerol trinitrate, and its profile is less favourable for treating angina than glycerol trinitrate. Amyl nitrite has become a recreational drug known as poppers, used to enhance sexual experience due to its vascular effects in the penis, but its vascular effects mean that it is a potentially dangerous drug to abuse. Nitric oxide is not the only modulator of smooth muscle function that is released from the endothelium. Another mediator is prostacyclin, or PGI2, which is a product of arachidonic acid metabolism by the enzyme cyclooxygenase. Prostacyclin is released from the endothelial cell and acts on the prostacyclin receptors on the membrane of smooth muscle cells. These receptors stimulate adenylate cyclase and the production of cyclic AMP, which relaxes smooth muscle as a consequence of the cyclic AMP-dependent phosphorylation of a number of proteins involved in regulating contraction. When the nitric oxide and prostacyclin pathways are blocked, activation of receptors on the endothelium still causes muscle relaxation, but the relaxation depends on hyperpolarisation of the smooth muscle cells. It was therefore proposed that there is an endothelium-dependent hyperpolarising factor which operates independently of nitric oxide and prostacyclin. This pathway is not yet fully understood, but several molecules have been proposed as candidates for the hyperpolarising factor. One of these is another product of arachidonic acid metabolism. The epoxygenase enzyme, cytochrome P450, produces epoxy-icosaptrienoic acids, or EATs, from arachidonic acid, which can then hyperpolarize the smooth muscle. Potassium ions and hydrogen peroxide have also been proposed as endothelium-derived hyperpolarizing factors. Potassium is released from endothelial cells by flowing out through potassium channels that are opened by the rise in cytoplasmic calcium concentration. Because the distance between endothelial and smooth muscle cells is very small, only a few nanometers, it has been proposed that potassium accumulates in the intercellular space to effectively raise the potassium concentration outside the smooth muscle cell. This shifts the equilibrium potential for potassium, resulting in potassium flowing out of the smooth muscle cell through inward rectifier potassium channels. That in turn causes hyperpolarization of the smooth muscle cell membrane and therefore muscle relaxation. Not all the factors released from the endothelium are vasodilators. Endothelial cells release endothelin peptides, the main one being endothelin-1. Endothelin-1 acts on ETA receptors on smooth muscle cells to evoke contraction. In some blood vessels, there are also ETB receptors on the smooth muscle cell, which also evoke contraction. Endothelin-1 released from the endothelium can also act back on ETB receptors located on the endothelial cells to evoke the release of endothelium-derived relaxing factors. This action of endothelin therefore interferes with its contractile action on smooth muscle. There are several drugs that act as competitive antagonists at endothelin receptors. Bosentan is an example of a non-selective endothelin antagonist, which inhibits both ETA and ETB receptors. Ambrisentan is an ETA selective antagonist, so preferentially blocks the ETA receptors on the smooth muscle cells. This endothelin system plays an important role in the pulmonary circulation, and high levels of endothelin are associated with pulmonary hypertension when blood pressure in the lungs is elevated. Endothelin antagonists have therefore proved beneficial in the treatment of pulmonary hypertension when they lower the arterial pressure in the lungs by inhibiting the action of endogenous endothelin. Endothelial cells can also communicate with smooth muscle cells via gap junctions. This slide illustrates how gap junctions are formed. The building blocks are connexins, which are proteins with 4 membrane-spanning domains and intracellular carboxyl and amino ends. 6 of these connexins come together to form a connexon, or hemichannel. Connexons can be homomeric, in other words, they are made from the same 6 connexins, or they can be heteromeric, with more than one connexin contributing. Gap junctions form between connexons expressed in 2 neighbouring cells, and they provide a channel that spans the membranes of both cells, connecting the interiors of the 2 cells. Each cell may contribute the same connexin to the gap junction or different connexons. When the gap junction is formed by the same connexons from each cell, it is said to be homotypic. Where the connexons contributed by each cell are different, the gap junction is heterotypic. Gap junctions can contain homomeric connexons in each cell, or one or both connexins can be heteromeric. This diagram shows how 2 cells communicate via gap junctions. It starts when ionic current flows across the membrane in one cell due to the opening of ion channels. The current generates a change in the membrane potential, which is conducted through the gap junctions into the neighbouring cell. So the membrane potential of the second cell is changed, and that can result in a change in the activity of ion channels in the second cell. In the blood vessel wall, gap junctions are found between pairs of muscle cells or pairs of endothelial cells. They are also found to connect endothelial cells with muscle cells. Although the endothelium and smooth muscle cells are separated in the artery wall by an internal elastic lamina, the elastic sheet is not entirely continuous. It contains small holes through which endothelial cells can project and come into contact with a smooth muscle cell. Where this occurs, the cells can communicate through gap junctions. Gap junctions between smooth muscle and endothelial cells are known as myo-endothelial gap junctions. In some blood vessels, gap junctions are thought to mediate the endothelium-dependent hyperpolarisation and relaxation. Activation of the endothelial cell raises the cytoplasmic concentration of calcium, which opens calcium-activated potassium channels. As a consequence of potassium ions flowing out of the cell through these channels, the endothelial membrane hyperpolarises. The hyperpolarisation can be conducted through myoendothelial gap junctions to cause hyperpolarisation in the smooth muscle cell, which leads to muscle relaxation.